Uh-huh. Uh-huh. Okay, so morphology. There's pleomorphic. We are talking about influenza virus. Morphology of influenza virus, they are pleomorphic, appearing spherical or tubular, enveloped RNA viruses. All you have to know about viruses. I gave you scheme of answering. Viruses answering what shape they've got, what type of nucleic acid, and is this virus naked or enveloped? Most pathogenic viruses are RNA viruses. Just a few viruses you will study. Really, just among pathogenic viruses, the only 6 genera viruses have DNA. And by our program, we are not going to study all of them. You will study herpes viride, herpes viruses, their DNA, and hepatitis B virus. So memorize 2 viruses. Pathogenic viruses are DNA viruses. You will study all the viruses RNA, and most of them are enveloped viruses. Most of them. It's essential information you should know. There is a photo of electron microscopy, real influenza virus in electron microscope, maybe spherical, maybe a little bit tubular shaped. And do you see spikes on the surface? It has envelope, and the spikes go through envelope. This structure, influenza virus structure, just picture. Inside of every virus has a nucleic acid inside, in this case RNA, and there are internal proteins, so nuclear protein, and it is surrounded by capsid. This green color means capsid. Capsid consists of proteins, M proteins, and it's important. And capsid is called by envelope. About the capsid, this is lipid envelope, and viruses have no own lipids. Anyway, there are lipids in envelope, and they are not viruses lipids. They are substances of the host cell. After replication, viruses go out slowly, and really every virus consists of nucleic acid and capsid only. But if viruses, ready virus particle, go out from the host cell after reproduction, they are carved by the substances of the host cell. And anyway, they have spikes. Most of viruses have spikes. Spikes go through the envelope, and they are glycoproteins, own viral glycoprotein. They have viral origin, and even the capsid is carved by envelope, where foreign substances or human being substances. Anyway, spikes are on the surface, and there are some spikes have functions of neuraminidase, sialidase. They are able to destroy sialic acids, and some of them, red color in this picture, hemagglutinins. Hemagglutinins are HA, neuraminidase, NA, different, and they are different. And of course, they are specific viral antigens. The envelope contains 2 glycoproteins, GP hemagglutinin, HA, and neuraminidase, NA. Functions of the viral spikes. Hemagglutinin function, HA. It is the viral attachment protein. It helps virus to attach to the surface of the host cell. Then it promotes fusion of the envelope to the cell membrane. It hemaggglutinates. What does it mean? Agglutination means aggregation. Hemagglutination means aggregation of red blood cells. So by using of hemagglutinin, viruses are able to bind and aggregate RBC. Of course, finally, it leads to destruction of blood vessels, and it leads to bleeding. So it's a very important virulence factor, and most of pathogenic viruses have these hemagglutinins, most of them, and also has protective neutralizing antibody response, or this hemagglutinins, and neuraminidase functions. It is enzyme, neuraminidase or sialidase. It cleaves neuraminic acid or sialic acid to release progeny virus from the infected cells to help go out. It also degrades the protective layer of mucus in the respiratory tract. This enhances the ability of the virus to infect the respiratory epithelium. Hemagglutinin functions at the beginning of infection, whereas neuraminidase functions at the end of replication. And hemagglutinins and neuraminidase are on different spikes. And of course, they are antigens of specific viral antigens because of in diagnosis of viral infections, of course, identification is done. The same like in case of other infectious disease, the specimen is sent to the virological laboratory with presumptive diagnosis. And the aim is to find the presumptive causative agent. So virus should be identified. I'm repeating, identification is a detection of species name. What species these viruses belong to? Do they belong to the species of presumptive causative agent? And viruses have no so many properties like other microbes like bacteria, and viruses are identified just in accordance with antigenic structure. So antigens are very important, of course. Every substance of virus is antigen, and even nucleic acid, but it is weak antigen. Antibodies are produced, but in diagnosis they are not detected. Capsid proteins are very important; they play a very important role, and hemagglutinins and neuraminidase are specific viral antigens. What this picture? Influenza A virus replication. This is the host cell, human being cell. And this is virus. This is red color RNA, green color envelope, spikes. So a virus is able to attach to the surface of the cell. Then by endocytosis, virus go inside, uncoating. So envelope is destroyed, just RNA is necessary for replication. Then a replication of viral RNA, transcription, and then viruses reproduce very unusual way. Replication of nucleic acid is going on at one part of the cell at nucleus, you see, and viral proteins are produced at another part of the cell at another time. So proteins are accumulated at some part of the cell, and nucleic acids. And what the human being cell with replicated viruses inside is named infected cells. So in infected cells, it is possible to detect aggregation of proteins of virus they are produced, and some aggregation of nucleic acid. And then assembly happens. Nucleic acid is carved by proteins. And now viruses are ready, maturation there. Now they are ready. And ready viruses go slowly, so they are carved by the cell wall of the host cell. They will get envelope, and viruses go out. It's important, I especially show this picture, to realize that in infected cell, it's possible to, even by using off-light microscope, if we will stain the cells from the human body, it is possible to find some unusual particles in infected cells, and they are named inclusions body, and it's important for diagnosis. Just microscopic honesty is used, of course, and it is better to use electron microscope, of course, but if there are no electron microscopes, it is possible to do microscopy by using off-light microscope for detection of inclusions body. Detection of them indicate that the cell is infected. It is indication method, not possible to identify, but it is possible to realize that the cell is infected by a virus. It is possible to detect inclusions body in the specimen from the human's body, and it is possible to detect the same inclusion body if viruses are cultivated in cell culture. we are looking for the same inclusion body. Cultivation, how it is possible to cultivate virus. Just a bit. Cultivation. Memorize that viruses don't grow on artificial media. All viruses, without any exception, are obligate intracellular parasites. So they don't grow on artificial media and they need the host cell for replication. 3 methods are used for cultivation of obligate intracellular parasites. I hope you remember from chlamydia. We did study bacteria, chlamydia, but they are obligate intracellular parasites. So I told you about these 3 methods. Number 1, the best method, cell culture. Different cell cultures are used, isolated from animals or human body, and not necessary to memorize exactly what cells, just cell culture. And if it is a monolayer of the cell, it is cell culture, maybe multi-layer, tissue culture it is named. Anyway, the cells culture have own names like HeLa, Hep2, monkey kidney cells, embryo human kidney, and so on. Not necessarily to memorize what are they, just cell culture in general. Then the second method is an embryonated, hence an egg. In short, it is named Chicandria. And it should be embryonated because people use for food an egg that is not embryonated. In this case, we need an embryo inside. And laboratory animals, they said, for every virus, the proper cultivation method should be should be detected, because of it. It doesn't mean that any method is used for cultivation of any virus. For example, hepatitis viruses, they are able to reproduce in very special cell culture, like cancer cells, liver cancer cells, and possible to notice reproduction. So for them, no methods for detection except laboratory, except animals such as apes, chimpanzee are sensitive to hepatitis viruses. It was detected, but of course it's not convenient to use apes in practice. They are used just for scientific purposes. So it depends. Influenza virus, for cultivation of influenza virus, all 3 methods are possible to use. And cultivation in cell culture, there is just picture how it is supposed to take some organs from animals, maybe from the chick embryo, and then by using of enzyme, they should be separated, these cells, and then we have to place some medium in special bottle or maybe petri dish, maybe some polystyrene plate. Cells need substances and they should be alive. So viruses don't grow on media, but we need a medium to create for metabolism of the cell culture. They should survive. And primary cells, they are able to exist very small, a short time. Then maybe diploid cells, specialist cells, those are able to exist through 3 inoculations and approximately 50 times. And then cancer cells. Cancer cells are able to multiply constantly during many decades. And so different cell cultures are used. And then how it is looking, cell culture method. It is looking like, so maybe special bottles. Exactly, the cell should be located on this side of this bottle, and the color is medium, that is necessarily for these cells. Maybe petri dishes, plastic. No, everything is plastic, of course, disposal on time using. Maybe petri dishes, maybe polystyrene plates, and there is medium that is necessarily for metabolism of the cells. And inoculation of the specimen in the cell culture is done. We should place the cells on this bottle, and then we should place some specimen. before inoculation, we had to examine by using of light microscope, and we had to see the monolayer of cells. And after inoculation of specimen, we should again examine this cell culture to find some changes for indication of virus. Now, the second one method, inoculation of specimen in Chicandria. There is scheme, this is embryo, and different parts of chick embryo are used for inoculation. This is air sac. Of course, no inoculation is not done into the air, and amniotic inoculation, a yolk sac inoculation. This is yolk sac, yellow color, baby allantoic inoculation, and sometimes chorioamniotic membrane inoculation. For 13 virus, 13 part of chick embryo is used. For influenza virus, amniotic inoculation, not necessarily to memorize, but realize it's not simply. For every virus, the 13 part of chick embryo is used. And the third method is laboratory animals. Different laboratory animals are used, like apes, like mice, and inoculation of the specimen into the nose of mouse. Because of the portal of entry, virus influenza viruses are transmitted by droplets, and the portal of entry is mucosa of nasopharynx. So the specimen should be injected exactly into the nose. In case of other viral infections, other specimen is inoculated maybe intravenously, maybe into the brain, like for Russian spring summer encephalitis virus, it has affinity to neurons, so the specimen should be inoculated exactly into the brain. So the method of inoculation depends on the virus. In case of acute respiratory infections viruses, of course, viruses should be injected on the mucosa of nose. So cultural properties. Now, antigenic structure, antigens, very important. Please pay attention, because of due to very changeable antigenic structure, the influenza is still a very big problem all over the world. I'm repeating, in spite of many vaccines, a lot of vaccines are produced and they are used every year, but no one effective till now. Why? Because of influenza viruses are classified as types A, B, and C, and and there are 3 genera: influenza virus A, influenza virus B, and influenza virus C. So, and they are divided on types depending on the inner proteins that are inside, mainly M protein, they are named, and NP proteins, then there are F proteins, other proteins. So influenza viruses are divided on 3 types, A, B, and C. And then only type A includes subtypes. I hope you believe, you know from bacteriology, if strains of some species of bacteria differ by antigenic property, they are named serovariants, serological variants. And if there are many serovariants, they are joined in serogroups. Like if you remember, Leptospira, for example, approximately 27 serogroups include 200 serovariants. Salmonella, more than 67 serogroups includes 2,500 serovariants, and so on. Before the term variant type, the term type was used, and it's still used in virology. So if we are talking about type and subtypes, we are talking about strains of viruses. Those belong to one species and differ by some antigenic structure. Okay, so type A includes subtypes, and the hemagglutinin and the neuraminidase are the subtype-specific antigens. And the antigenic variation are caused by hemagglutinins and neuraminidase, which lead to the serological subtypes. Human and animals influenza viruses, you know, these influenza viruses are circulated between people, between animals and birds, And in general, there 15 hemagglutinases are detected, like 1, 2, 3, and so on, 15, and 9 neuraminidase, but completely. And subtypes of influenza virus are designed by combinations of hemagglutinins and neuraminidase. For example, type A, H1N1, for example. And completely 15 between human and animal influenza viruses have got 15 hemagglutinins and 9 neuraminidase. And just 4 hemagglutinins were detected, were isolated from human beings, 1, 2, 3, and 5, and 2 neuraminidase, 1 and 2. Anyway, variations of hemagglutinins and neuraminidase organize subtypes. And nowadays, okay, subtype A, type A, for example, subtype H1N1. And now we are talking about group of viruses. Those have hemagglutinin number 1 and neuraminidase number 1. But inside this group, viruses are differ again by proteins, antigens. And again, there are differences. It doesn't mean that all strains, those have hemagglutinin 1 and neuraminidase 1, have similar antigenic properties. There are differences by proteins, antigens. And nomenclatory systems include information, serotype, host of origin, geographic origin, strain number, year of isolation. Like, for example, A means type A, Hong Kong, place of isolation. 01, strain number, year of isolation, 68, and then subtype H3 and 2. Well, this is characteristic of influenza viruses, international characteristic. Antigenic variability of influenza viruses. Why I'm repeating, don't forget, antigenic virus has very changeable antigenic structure. And many strains of influenza virus have different antigenic structures. There are two mechanisms of antigenic variability. Number one, relatively small changes, antigenic drift, and strong changes, named antigenic shift. Antigenic drift occurs between pandemics in all types of viruses. These are minor changes in the structure of surface antigens, hemagglutinin and neuraminidase, caused by point mutations in the gene that encode them. Changes happen annually, maybe in 2-3 years, and the result is an epidemic. Because of, again, new serovariants will cause epidemic. People have no immunity to realize if strains of species of microbes are differ by antigenic structure. The character of immunity is very, very narrow, exactly for every antigenic variant. And so reinfection are very often. Species immunity is not developed. So epidemics, many people again are infected, and most of them develop clinical symptoms. Antigenic shift are major antigenic changes resulting from the reassortment of genomes between different strains, including animal strains. But this occurs only with influenza or influenza A virus. So at irregular intervals, from 10 till 40 years, viruses appear with strong differences. The antigenic structure of hemagglutinin and less often neuraminidase changes. But there is a recombination of genes of the influenza virus of animals and humans. The influenza virus contains 8 RNA molecules, and if 2 different influenza viruses are found in the same organism, an exchange of nucleic acid takes place. So by this reassortment, have you ever heard about swine flu and then birds flu, and such epidemics were developed exactly due to genetic recombination between influenza viruses of human beings and animals and birds. Serotypes are most dangerous for humans. Serotypes AA viruses can change their antigenic structure and cause pandemics. For example, bird flu virus AH5N1. It can also cause human diseases. The outbreak was detected, and now time by time epidemics are detected in different countries. Last year in Russia, in the south of Russia, on some birds farm, the influenza was detected between hens, and the virus was transmitted to the human beings. They were infected by this bird's flu virus. The internal ribonucleoprotein is the group-specific antigen that distinguish influenza A, B, and C viruses. There is a picture like reassortment, avian virus, human viruses, and then pig, they did meat, and pandemic flu was developed. Avian, human, reassortment, inpatient, again, pandemic flu. Then avian, chicken, mutation, and inpatient body, they were reassortment and pandemic flu. So from the wild birds, domestic birds were infected, and now people are infected. So influenza virus is circulated among animals, birds, and human beings. Now some strains are common viruses, okay? This is picture, shows what, how it is developed. And many species of animals, like, for example, aquatic birds, chickens have their own influenza A viruses. These animal viruses are the source of the new antigenic types that cause epidemics. And for example, like swine flu was caused, and the same symptoms of swine flu were detected, like in case of influenza: systemic fever, high temperature, the lethargy, lack of appetite. In nasopharynx, runny nose, sore throat, respiratory, coughing, intestinal diarrhea, and gastric nausea and vomiting, and even the clinical symptoms of the some problems with intestinal tract are developed. I'm going to make a break, and then I will send you the link again. 10 minutes break. Do you hear me, leaders? And then I will send you the
Yes, doctor.
link again
Okay.
in 10 minutes.